everyone, how are you? I hope you all are fine. So in this video, in previous video, we have discussed about difference between plant cell and animal cell, and about difference between prokaryotic cell and eukaryotic cell. And in this video, we will discuss about cell modification. Okay. Means how are the cell enveloped? Enveloped means things that cover the cell. It means enveloped. Means enveloped in the with the help of envelope we take here later for safety. In the same way, envelope means to protect the cell. Which type of envelope are present for its modification? So let us discuss about this. So here our first point is, most prokaryotic cells, particularly the bacterial cells, have alchemically complex cell envelope. It means most of the prokaryotic cell, particularly means the bacterial cell have a chemically complex cell envelope. Means without any chemically, they have a complex, means very hard type cell envelope present. Okay, the cell envelope consists of a lightly bound 3 layer. Which type of layer? 3 layer the cell envelope are consist. Like outermost is called glycocalyx, followed by the cell wall and the plasma membrane. It means, suppose this is the glycocalyx. Okay, this one is so this outer layer is called glycocalyx. Okay, and followed by which? Cell wall. It means after that the green color is what? Cell wall. There are no between space between glycocalyx and cell wall. I have made this because you can understand which is glycocalyx and which is cell wall. Okay. And the third one is plasma membrane. It means the innermost layer is called plasma membrane. This type of cell envelope are present in prokaryotic cell. Okay. Outermost layer is glycocalyx. Okay, then the inner green was sorry. Okay. So according to this sentence, we got that outermost layer is called glycocalyx and followed by the cell wall and the plasma membrane. Okay, this was the envelope of which cell? Prokaryotic cell. I think it is understandable. Our next point is, glycocalyx is the outermost layer. We have seen here that glycocalyx is outermost layer. Okay, and comprising a coating mucus or polysaccharides macromolecules which protect the cell and also helps in adhesion. It means the glycocalyx have mucus. Mucus means sticky like substance, or a polysaccharides. It is a type of a chemically made like macromolecules which protects the cell and also help in adhesion. It means we see here that glycocalyx have mucus. Mucus means sticky substance. That's why it also helps in adhesion. It means to stick together, to stick with another one. Okay. like this. It means if here is mucus is available, then other substance can easily come to here. If the glycocalyx have sticky substance, then anything or any particle can stick easily in the glycocalyx. Okay. Now here is this type of chemicals. Okay, protects the cell wall, and this layer differs. This layer differs in thickness and chemical composition in the different bacteria. Okay, it means if the adhesion of glycocalyx is the help in adhesion also, it means to stick one another, and its thickness and chemical composition in different bacteria. It means different glycocalyx function have different function in different bacteria. Okay, so these are the some points about cell envelope of prokaryotic cells. Okay, like outermost layer called glycocalyx, innermost layer called plasma membrane, and middle which one called cell wall. Okay, so let's discuss further. Here some have a loose sheath called slime layer which protects the cell from a loss of water and nutrients. It means some prokaryotic cells have slime layer. Okay, and which protect means the slime layer which protects the cell from the loss of water and nutrients. It means the loss of water was continuously followed by a cell. It to prevent from slime prevent from it. Okay, like loss of water and nutrients from the cell. Some may have a thick and tough covering known as capsule. It means glycocalyx is the thick and very sticky in nature, but some cells, prokaryotic cells, have a thick and tough covering called capsules. Okay. The capsule and slime layer are made up of polysaccharides. Polysaccharides is a chemical composition. Okay. So here is written that the capsule and the slime layer are made up of polysaccharides. It means polysaccharides made the capsule and slime layer. I hope you understand all these points about envelope of prokaryotic cells. So move further. Here we have some more points about cell envelope. So our first point is the capsule is responsible for giving gummy and sticky character to the cell. It means glycocalyx is also responsible for adhesion, and the capsule are also responsible for giving gummy. It means sticky. Gummy and sticky. Here is sticky already mentioned. And character to the cell. It means sticky character which is given by capsule and glycocalyx. It allows bacterium to hide from host immune system. It also allows that to hide from the immune system. Hide bacterium from the host immune system. Okay. Host immune system, it means WBC to it also helps to hide from the WBC, because WBC is host of the bacterium. It can kill the bacteria from the body. Okay, so it helps to hide from the WBC, immune system. The cell wall determines the shape of the cell and provide a strong structural support to prevent the bacteria from the bursting. It means cell wall. Here is all the functions of glycocalyx and capsule, and now this is the function of cell wall. What is the use of cell wall in prokaryotic cell? The cell wall is determine the shape. But it means it determines the shape of the cell and also provide a strong structure, support also, because cell wall is made up of hard brick like structure. Wall means thing that protects. Okay, in the room the wall also protect from the wall also give us shelter to for live, and for from and also prevent wind, also rain. Okay, in the same way cell wall also provide a structure support to provide the bacterium from bursting and collapsing. It means the cell wall is most hard things, and if the cell wall is not in the cell, it can be collapsed. It means they can be stick together like this. It means if the cell is like this, and after some time if the cell wall are not present in this cell, it can be collapsed like a small one like this. Okay, और from bursting it means blast होना एक होता है ना, उसी तरह से collapsing. Our third point is a number of antibiotic inhibits cross-linking of peptidoglycan and strands, therefore cell under glycolysis in the presence of this antibiotics. It means a number of antibiotic is like a which cut some disease, like if we have any allergies, we eat antibiotic of it, that can prevent it can reduce the power of reaction level. So inhibit the crossing linking of peptidoglycan strands, and therefore cell under glycolysis in the presence of these antibiotics. It means cell do under glycolysis in the presence of which type of number? It is the number of antibiotic. Okay, the number of antibiotic inhibits cross linking of peptidoglycan. It means peptidoglycan inhibits. It means to stop the number of to cross antibiotic cross the linking of peptidoglycan. It means the linking by the peptidoglycan can be crossed by antibiotic. Okay, our next point, this is not a point, this is a new topic, like this one is an example of gram staining. Okay, so what is this? Let's discuss. What is gram staining? This is a new point. You have to know that it is a new term. It is a special technique in which he used to classify bacteria into two groups, gram negative and gram positive. This one is also used for to classify bacteria. It means the bacteria into gram staining and gram positive. Gram staining is the special technique to divide the bacteria into gram staining and into gram negative and gram positive. Sorry. And what is gram staining and what is gram positive? Let's discuss. Those bacteria that take up the gram stain are called gram positive. It means those bacteria can take up the gram stain called gram positive, and those bacteria who don't take up the gram stain called gram negative. Okay, so this one is the point of gram staining. Gram staining is nothing, it is only a process to classify the bacteria. Okay, the plasma membrane is a semi-permeable in nature. It means semi-permeable means not permeable. Permeable means to allow all the things. Semi-permeable means to allow something and some particular things. Okay, in nature, and interacts with the outside world. Okay, this membrane is a similar structurally to that of the eukaryotic cell. Eukaryotic. It means eukaryotic cell have the semi-permeable nature of plasma membrane. Okay. And our second point is a special membranous structure is the mesosome, which is formed by the invagination of plasma membrane into the cell. We have seen the mesosome, like see the structure of the mesosome. Like this is the outer layer of bacterial cell, and this one is inner layer of bacterial cell. Okay, which one is cell wall and this one is glycocalyx, and the water is the most inner layer called plasma membrane, and this layer perform the finger like innervation like this. What is this called? What we call this? I have explained in the previous class also. This is called mesosome. Okay, acts as mesosome acts as which? Mitochondria. Okay. So here is the point. A special membranous system. This is 2 points are about plasma membrane. Okay. So is this point is related to eukaryotic cell and this point is related to prokaryotic cell. That's why here is written that a special membranous structure is the mesosome, which is formed by the invagination. Invagination means a finger-like structure of plasma membrane in the cell. Here we can see that in prokaryotic cell, outermost called glycocalyx, innermost called cell wall, and most inner called plasma membrane. And we can see here that plasma membrane, the plasma membrane invaginate in the inner like this, and this perform like a mesosome. Okay, here is not a single membrane are touched with a cell wall. This one is green one, it means the plasma membrane. This green part is called plasma membrane, it means cell membrane. Both name are of plasma membrane. Okay, this point is, I hope you can understand this point because I have explained this point in better way. So here are more about plasma membranes. So let's discuss. These extensions are in the form of vesicles, tubules, lamella. It means these extension. It means the extension of plasma membrane in the form of lamella, tubules, and vesicles. Okay, we will study in ER and in lamella. It means in plastic. And about tubules, it is also come in ER in the sense endoplasmic reticulum. They help in the cell wall formation. It means the extension which is formed by the plasma membrane help in cell wall formation and DNA replication and distribution. DNA replication we can't understand in this video. We have to read. There is a chapter of DNA replication in which chapter? In cell division. Okay, and distribution to daughter cell. It means this all point you have to learn in the cell wall. Division of cell wall, you can understand there better. Okay, so move further. They allow help in the respiration secretion process to increase the surface area of the plasma membrane and enzymatic constant. It means they also help in respiration secretion. It means in the mesosome we have seen that here is a mesosome, and this is a formation invagination of plasma membrane, and here is the dotted like structure called respiratory enzyme. Well, in the same things are written here that respiratory secretion They also help in respiration secretion process to increase the surface area of the plasma membrane. To increase the surface area. If the finger-like structure is formed by the plasma membrane, it can increase the space of plasma membrane. So, and the enzymatic constant. Why same cheese? Same thing are written here. The enzymatic constant, it means the respiratory constant are in the constant manner, means respiratory enzyme are in the constant manner, means in the number of, they are in the number, not in infinite manner. Okay. And here is one more point is about mesosome is found in gram-positive bacteria. Mesosome is only found in which classify the bacteria in two parts, gram-positive and gram-negative. And here mesosome is only found in gram-positive, okay, not in gram-negative. In some photosynthesis prokaryotic like cyanobacteria, purple bacteria, these are the other membranous extensions into the cytoplasm called chromatophore, which contain pigments. Chromatophore, I have made the one figure like is like, and here I have made a like this, like nucleus like a structure, and I have called them vector. Vector is also called chromatophore. Okay, this contain pigment like a plastid. They help into photosynthesis. Okay. So this is all about the prokaryotic cell envelope and in its modification, and it also we have learnt about some plasma membrane modification, like plasma membrane modified into mesosome. Also they can also in the form of lamellae and tubules also. Okay, so this is all about in this video. Okay, and in next video we will start flagella, and we will read about flagella, and we also read about some overview of cell. Okay, and the introduction class is finished. Okay, and after that we will start cell wall. So be careful, take care, goodbye.